on this computer. Is it recording? Yes. Okay, so there's a very important document for paper 5. This is your notes for paper 5. So it's a very important document that you want to refer to know exactly what you need to know every time when you do the passive questions. Okay, always come back to here. So this is called a paper 5 overview, right? So I hope all of you have already printed it out, or else you have already uploaded to your apps. Okay, so so paper 5 is planning, analysis, evaluation, and conclusion, which I've already discussed with you just now, right? So before that, for paper 5, paper 5 is a higher order experimental skill. So you know that when you do experiment for science, whether it is biology, chemistry, or physics, when you do the experiment for science, you're actually following this method called scientific method. Okay. Right. So this is called a scientific method. So you're following a scientific method. What is scientific method? Is a way for you to approach to find out some information about the nature. Okay, you want to find out some information about the nature to see whether the nature behave in this way. Does the nature behave in this way? So, and then once you find out, okay, you can actually inform others about this. Okay, for example, for physics, there's a gravity there, and there's a formula for gravity. Okay, and then in biology, there's such thing called temperature affects enzyme, rate of enzyme catalyzed reaction. So how do we know that this is the nature of how things behave? In science, they always use scientific method to verify it. So every time when you do experiment, we're actually following a scientific method. So I'm going to go bring it through with you again, because you already learned it in your secondary school, but this is a very important foundation. So I will go through it again so that you know when you do experiment, actually what you are doing. Okay. So in science, we're talking about learning about the nature. We want to learn about the law of the nature, how things work in the nature. So there'll always be questions, okay? Question about how things work. So what does the scientist want to learn more about from the nature? So there's a question there. For example, does increasing light intensity will increase the rate of photosynthesis forever? So as you increase the light intensity more and more, does the rate of photosynthesis also increase more and more? So you want to know. So there's a question. Then you will go and do research. Do research means you want to find out, are there anyone before you who have done experiment and find out what you want to find out? So you have to do research first so that you don't have to duplicate the work when the conclusion is already derived by somebody else. So therefore you have to do a literature research. That means you read through all the journals which record the past scientist experiment journals, okay, to find out whether they have actually done any experiments pertaining to the questions that you have. Okay, so after that, you have done the research, you find out, okay, there are some related experiments, but there are no experiment that relates to what exactly I want to know. Then you can conduct an experiment, okay? You can use the research experiment and modify it to conduct your experiment. So before you conduct experiment, you must write your hypothesis, right? So your hypothesis is an educated guess. So hypothesis is what you want to find out. So your guess is, when I increase the light intensity, the photosynthesis rate will increase. So I'm guessing that this is so, okay? So your hypothesis is an educated guess. I guess that this is how the nature happens. So after that, you will need to, after hypothesis, you will need to write out your experimental procedure or method. So how are you going to conduct the experiment to find out what you want to find out? So you want to test your hypothesis. So you write your procedure and then carefully follow the written procedure to to carry out the experiments. Okay, so first you must write out your experimental plan. Then when you carry out experiment, the next step is you carry out experiment and you obtain your data. Okay, so your data is your result of your experiment. Okay, so your results can be qualitative or quantitative. All right. Then after you get your result, you have to analyze your data. So analyze your data means sometimes from there you have to do calculation. From there you look at trends. So when light intensity increases, does the rate of photosynthesis keep on increasing, or it increase until certain value and it levels off? Right? Okay, so look at the trend of the table and graph, and you also can do statistical analysis through your results. Okay, that means to find out whether your results is significantly correct or not. Okay, significantly correct or not. Okay, then you can do conclusion. So conclusion means conclude. Do the results support your hypothesis or negate your hypothesis? Negate means negative. Negative means go against, means it is against your hypothesis. Okay, it's not aligned with your hypothesis. So once you do your conclusion, you can remodify. Okay, let's say your conclusion says that, okay, yes, increased light intensity increase the rate of photosynthesis, but up to a certain level, then it becomes level off. Then you may want to modify and retest your hypothesis. So why does it level off? At this point here, what are other things which actually causes the rate of photosynthesis to become constant? It does not increase anymore. Why? So you can do another experiment to find out. Okay, so this whole process here is called the scientific method. Okay, so everything that you learn in science, biophysics, chemistry, all of them are actually found out based on the scientific method. So that we don't just say because we feel like it or because we think it is so, okay, we assume it is so, but we actually go and conduct experiment, okay, and in experimental condition, it actually shows that yes, it actually supports the hypothesis, so this statement is correct. So this is called the scientific method, okay? But you need to bear in mind that whatever we can find out about the nature is still limited by the instrument that we have, okay? For example, when there's no microscope, when you tell them that there are organisms that your eyes cannot see, nobody will believe you because I cannot see what, how can it be there, isn't it? So it doesn't mean that you cannot see or it's not proven by scientific method, it's not there. Sometimes we do not have the instrument to be able to measure the nature. So science is still quite limited, even though it is able to find out a lot of things. You need to know that it's limited by the instrument that's able to measure things. If you cannot measure that frequency that which exists will never be known to us. So you must have this in your mind. Don't think, and I would say, don't glorify science, okay? Don't glorify science as the God, because there are a lot of things that science still cannot find out. So there's still a lot of things to be found. So that is something that you need to bear in mind. I won't say science are not good, science is good, but to a certain extent, there are a lot of things that the science cannot find out, okay? Limited by the instruments available. For example, you say, are there ghosts or not? Okay, your eyes cannot see, right? Are there any instrument can measure that there's a presence of ghosts? It's a different frequency, okay? So that thing, it depends, all right? It's not about religion, it's about science. Okay, so that's interesting, right? So now I'm going to introduce you to paper 5. What are the things that will be tested in paper 5? So this table is obtained from, I'm going to increase the size so that's easy for you to see. So this table is obtained from the syllabus. So again, if you want to know very detailed, you want to have got very detailed notes, explanation for paper 5, you can go to your syllabus, just like your paper 3, right? Your paper 5 syllabus is this one here. It will tell you in very, very detail, every single part, exactly what you need to be tested. But most of them, you already learned it in your AS, so I will not go through it. I will use the notes that we have created for the students because they are really good and they're very, very clear cut, very, very clear. So I will prefer something that's very clear. You want to read more, you can go to your syllabus and read, okay? So in your syllabus here, it says that paper 5 is divided into two parts, into two parts. The first part is the planning. Okay, you need to plan the experiment, write out the experiment. So what is the problem? That means what is the hypothesis that you want to test? Okay, and then you write out the methods in detail. Write out your experimental plan so that somebody can follow your method to conduct experiment and get the results. So therefore, your method must be written in a way that anybody that take it can follow and get a very accurate, reliable results. Okay, so we'll go through that one for today. Now, the second half part of the paper includes analysis. That means when you get the results, how are you going to analyze the result? And evaluate, so evaluate the experiment. Okay, whether the experimental design is can give you accurate and reliable results. What are the areas that you need to improve on? Okay, is it the apparatus you need to improve on? The control variables you need to improve on? Replicates you need to improve on, and so on. Okay, conclusions. So conclusion from the results. So today we will look at the planning, which is also a revision for you because I've done it in AS. I've stressed it a lot in AS. So now for A2, you'll find it's very easy now for the planning part because you're already quite familiar with it. So I would like to spend the first part of today's session on planning, and the second part will straight away go into doing a past year question. For paper 5, if you want to do well, if you want to be very confident to do well in exam, you need to do as many past year questions as possible, starting from 2016 paper. Okay, 2016 paper onwards, because this is a new syllabus. Correct? Yes. Okay, 2016 onwards. The old one, the old one, the students have to go to the lab and conduct experiment one, whereas the new one is all theory. Okay. Okay, so let's go back to your notes. Okay, if you have, please bring out your notes so that you can jot down additional information. Now, this table is very good because this table actually outlines every single thing that you need to know about paper 5. Every single syllabus or learning outcome that is, they are all outlined here to show you these are the things that you need to know for paper 5. So it's very good summary. Okay, the syllabus does not have a summary table like this, only these notes have. Okay, that's why this note is very, very valuable. Okay, so the first part of the paper 5 is planning. So in planning, you know that there are 5 steps. Okay, there are 8 steps when you want to plan an experiment. Step 1, you must have a hypothesis, an intellectual guess. What is it you want to find out? Okay, and this hypothesis can be in words or in graph form. So we'll come to this one in detail later on. I'll outline first. So after that, you must have independent variable, dependent variable, standardized variables, and then you need to talk about detailed procedure. Then you need to talk about reliability precaution, safety precaution, and lastly, control experiment. So there are 8 steps that are important in planning. These are the 8 things that they will test you in the first half section of the paper. So very easy, isn't it? Okay. And then after that, for the remainder of the table, analysis, okay, and evaluation conclusion, I will not go through it yet until you need to learn it. So no need to see so many things yet, okay, because you're not going to learn it today. Just focus on planning, experiment will do. Okay, so very easy. So you try to make it's very important you know how to try to make your life easier for you. It doesn't mean that you're going to be lazy, but you know these are the huge chunk of things you need to read, but you can plan to read a small chunk per day. That's how you divide a very huge task into small portion, because small portion means you can digest, you can assimilate. So it needs time. It needs time for you to do that. So you need to start early and do consistently. Okay. Okay. Success is not doing a big time at one go, but it's doing a small task consistently every day. Okay, that's called success. If you can do a small task every day consistently, that is called the miracle and success. Okay. Okay. Right. So let's go and look at what are the 8 steps in planning an experiment. Okay. After today, you can already secure 50% of the paper, which is good, isn't it? And you've got 3 months to practice doing the questions. So if every week, 1 paper, you've got 3 months already have got at least 12 papers. Only 12 papers. If every week 1 paper, right? Only 12 papers, not a lot. Okay, just nice only. It's only just nice. Okay. All right, so let's look at first one. When you want to plan experiment, first thing is step number 1. You need to identify the hypothesis. Okay. Okay, I'm going to call your name to answer the question. So can you please be ready? Okay. Gen Z1. Right. Okay. Okay, now, when you want identify hypothesis, hypothesis is a statement of what you want to find out from the experiment. It's an intellectual guess. You want to find out something experiment. So in your hypothesis, you must have 3 things. Okay, first, you must have your dependent variable. Second, you must have your independent variables. And thirdly, you must have got a statement that connect the relationship between these 2. Okay, so how is independent variable connected to dependent variable? How does the independent variable is being connected to the dependent variable? Okay, how are they connected to each other? So this is what you need to have in your hypothesis. And the hypothesis can be in 2 forms when they ask you in the question. It can ask you to describe in words, or they can ask you to plot in graph. So there are 2 ways they can ask you. So let's look at the first example. This is a hypothesis statement. As the light intensity increases, the rate of photolysis of water will increase. So this is a correct hypothesis statement. Okay, can you tell me in this statement, right, which one is the independent variable? Alicia?
The independent variable is the light intensity.
Okay, very good. Can you tell me, so which one is the dependent variable? Chi Kit Ming? Chi Kit Ming? Is he here today? Kit Ming, are you here today? I think your class is full today, right? Because there are 31 of you here. Chi Kit Ming, but I don't see his name here. What happened? He's the only one not here. Maybe, huh? Okay, what about, you can see Tammy Lee Lok Wei. Tammy? So what is the dependent variable here?
The rate of photolysis of water.
Fantastic. All of you are on your way to score A star already. Very good. Okay. So you got your independent variable, you got your dependent variable. So tell me, what is the information that connects between this independent variable and dependent variable? Aidan Hall?
Light intensity increase, rate of photolysis of water increase.
Yeah, so this one, isn't it? So when light intensity increases, the rate of photolysis of water will also increase. So this is you're showing the relationship between the 2. Okay, so in a hypothesis, it tells us what you're going to test in your experiment. I'm going to test that, I'm guessing that the light intensity increases, rate of photolysis of water will increase. I'm guessing, so intellectual guess. So I'm going to test this in my experiment. So therefore, in your hypothesis, you must write both independent variable, dependent variable, as well as the relationship between that. Okay, Kit Ming is coming in now. Okay. Now, but if you're asked to write the hypothesis in the graph form, sometimes they give you an empty graph and they ask you to write. So do you know how to write this one then? You need to draw, isn't it? You need to indicate what is your independent variable and dependent variable, and then you need to show the relationship. So based on that, that means the relationship must be this way. Okay, or this way also can, or this way also can, because increases, isn't it? Right, so either way, but you must use your theory that you've learned to answer the question. Your theory is this one, so therefore the only answer is this one. It cannot be this one, it cannot be this one. That means even though it's increased, but based on theory, what you have learned about the nature of increase, is it a straight line or is it a curve? Okay, it's actually a straight line. Until maximum, it will level off, but you cannot draw this because it's not stated in the hypothesis. Okay, yeah, so I hope that's clear for you. Now let's look at the second example. As temperature increases, the rate of respiration increases. All right, so I must remember to write. So for this one, as temperature increases, oh no, this cannot be temperature. So this one is light intensity, and this one is the rate of, cannot write already, why? Rate of photolysis of water. So remember, you have to write the full sentence that is already here. Okay, so this is how, as we learn day by day or every lesson, this is how you learn to collect the marks. Remember, every 1 point that you learn, you add 1 more marks into your paper, right? So that you can score as high as possible. That's my wish for all of you, okay? My wish for all of you is you can score as high as possible. How high? The sky is the limit, okay? Okay, the sky, go and touch the sky, cannot touch, right? So the sky is the limit. That means infinity, okay? Okay, number 2. Step number 2 in planning experiment, you need to identify independent variables, okay? And then how to set it up, very important. So say for example here, so your independent variable is a variable that you deliberately change, or another word for it means manipulated variable. That means you deliberately change it, you change the temperature, you change the enzyme concentration, you change the light intensity. All these things that you purposely change, you deliberately change in experiment is called the independent variable. And you must always give 5 values to your independent variable, okay? You must always give at least 5 values so that when you got 5 values, you plot the point, you know whether it goes up straight or it goes halfway and then it turns. All right, if you only got 3 values, 1, 2, 3. Let's say you got 3 values here, you got the first point, you get the second point, and then you get the third point, then you can plot the graph. But you got the first point, second point, third point, you only get this shape. You wouldn't know that the shape will actually become horizontal later on. So when you got the 5 points, it's better. Okay, you may know the third point, the fourth point might be here. You do not know. Okay, so therefore it's best to have 5 values minimum. And it's always presented on x-axis or first column of the table. All right. So therefore, when you write in your planning, not only you must tell me what is your independent variable you're going to use, what is it, and what are the values, plus what is the unit. So this is what you need to give. So what is it? And you must give the 5 values that you're going to use, okay? And then you must give the units, okay? So this is your independent variable, okay? So next, you're going to look at your dependent variable. So dependent variable is something that you measure in the experiment. It's called dependent because it depends on your independent variable. It depends on your independent variable. That's why it's dependent. Okay? So this is something that you want to measure. A variable that may change depending on your independent variable. So this is your result of the experiment that you will record, or your observation. Okay? So usually you will, what you do is your independent variable, you need to either measure it, you want to write it down, either you measure it, so it is quantitative, okay, or you observe it, so it is a qualitative, right? Your result can be quantitative or qualitative. So you measure it, you got a number, you observe it, you can see color change, okay? Color change, or observation of the structure of the things, okay, or the change of the structure of the things. All right. Usually in experiment, it's better to get numbers as your data than observation. That means quantitative is better than qualitative. Because quantitative data, right, can give you more information, because quantitative data, not data, you can actually do calculations, right? You can calculate mean, you can do statistical analysis to know how accurate and reliable is your result. You need to do statistical analysis. Okay, so quantitative data, you can do all this calculation. Qualitative, you can't. You cannot calculate mean for color, isn't it? So because of that, you cannot do statistical analysis. That's why it's better to have quantitative compared to qualitative data. Okay. So for your result, you need to describe how you're going to measure it, okay, and record it down. And then, do you need to use a formula or not? After you measure, do you need to use a formula to calculate, to derive to your answer? If yes, how do you do your calculations? And after you calculate, what is the unit you're going to present it as? Okay, so I'm going to give you an example for this so you know better. Let's say, for example, you want to measure the rate of respiration of yeast. So if you're talking about a reaction, always C6, always write down the equation first. So C6H12O6, I want to look at the respirations in it. So plus 6 oxygen to form 6 CO2 plus 6 water. Okay, so this is a reaction that I want to look at. So you know that in an enzyme catalyzed reaction, there are substrates and there are products. Okay, and then you have learned from your AS that when I want to measure the rate of reaction, there are 2 ways I can measure, right, based on the graph that I have learned, right? 2 ways. So either it is substrate concentration versus time. I type is faster. Okay, substrate concentration versus time. Okay, time, or product concentration versus time. Okay, correct. So when the reaction starts, my substrate will be converted to product, so its concentration will go down, and my product will start to be formed, concentration will go up, right? So this is the curve that is in the graph. So you have learned that if you want to measure the rate of reaction, you either measure the rate of disappearance of product, which is rate of change in substrate concentration versus change in time. Okay, and then for product, so the rate is equals to rate of change in product versus time. So this one you are very familiar, right? How come got so many things here? Okay, yep. So when you are given an experimental apparatus to measure the rate, you may measure it differently. So your formula could be different, your calculation could be different, your unit will be different. Okay, say for example, you want to measure the rate of respiration of yeast. Okay, so you put the yeast in the test tube, not test tube, you put the yeast in the test tube, okay? Put the yeast in the test tube, and then you have got another tube, okay, where you transfer. So you've got a test tube with a bunk, okay, and you have got a tube to transfer the gas into another test tube, okay. Okay, it's already tilted, so I'm going to draw my test tube tilted as well, okay. And then I've got water inside this, okay. So I got my yeast mixed with glucose here, all right, and then I allow the yeast to respire and the gas is released. Okay, the gas is actually the CO2, and then it will be released here, and then the bubbles will be released. So in this kind of setup, when I want to measure the rate of reaction, I will actually measure the number of bubbles released per unit time, right? So I'm measuring CO2. So my formula now for this reaction is number of bubbles per unit time. So you know that you need to fix the time and measure the product in this case. Sometimes you can fix the product and measure the time. Time taken for all the substrate to disappear, for example. So in this case here is a number of bubbles per unit time. So you need to know what's the formula for you to calculate. After you calculate, what is the unit? Okay, I'm going to ask you, so if I'm calculating the number of bubbles in 1 minute, so what will be the unit that you will give for your result here? Annabelle? Annabelle? Is Annabelle there? Annabelle is here, okay. So Annabelle, what is the unit for this formula?
Sorry, Miss, could you repeat that?
Again?
Could you repeat that, please? I couldn't hear you clearly.
What is the unit that you're going to give for this formula? Yeah.
Is it seconds?
Seconds. Number of bubbles per second. Yes, you can put per second if it's very short time, or else you can put per minute. Okay, so because it's number of bubbles per minute, you can put per minute. If it's very short time, you can put per second. So it's correct. Okay, so therefore you cannot assume that the unit must always be cm cubed per minute because you're measuring the CO2 release. It must depend on the apparatus that you use and what you measure. That one you must be very careful. Okay? All right, so this is for your dependent variable. Take note of what you want to measure for the rate of reaction for the apparatus. What can you measure? So what is the formula that you can use to convert what you measure into the rate? Do the calculation and what should be the unit based on what you measure. Okay? So the next one, thank you, Annabelle, for your answer. Okay? So number 4, you need to identify the standardized variables and how do you control it. Okay, so so in your plan you need to identify, plus you must tell the person, okay, this is the thing, this is how you need to control it, so that the person can take your plan and do it very well. So what are the things you may want to control? Temperature, use thermostatically control water bath. pH, you can add buffer solution will control the pH at a constant pH. For concentration or volume, you must control the concentration and volume. Okay. For light intensity, now there are many ways to control light intensity. You want to have same light intensity, must be same distance of the light from your apparatus. Let's say, for example, you got a test tube and you got a unicellular photosynthetic organism in it, cholera. Okay, a lot of them. Then you got your light source here. Right, this is your light source. So when you got a fixed distance of the light source to your apparatus, this will give you the same light intensity. So make sure that every time you conduct experiment, you have the same distance of the light source. Or you can use a same voltage of light, okay, with the same distance. Or you can change the number of lamps. Let's say I want to do an experiment with 5 different light intensity. So for the light intensity, you can write 1 lamp, 2 lamp, 3 lamp, 4 lamp, 5 lamps. Okay, number of lamps, 1, 2, 3, 4, 5. Also can. Or you can use filter density. Filter density means you know the muslin cloth that you use. Okay, the muslin cloth that you use have got a lot of spaces in between to allow the light to pass through, right? So you can use 1 layer of muslin cloth that will block some of the light there. So you're going to put your muslin cloth maybe here, hold it here. Okay, use the instrument to hold it. Okay, or you can wrap it around your test tube also. Okay, so use 1 layer to wrap around the test tube, and then 2 layers, 3 layers, 4 layers, 5 layers. So that will give you different light intensity. Or you may want to change the distance of the lamp away from your apparatus also can. Okay, because the light intensity I, okay, formula, right? Light intensity is inversely proportional to, I mean, the L, let me write the L here, L, distance, okay? Light intensity is inversely proportional to distance. You increase the distance, you will decrease the light intensity, okay? Now for organisms, sometimes in experiment you use organism. So every time you use organism, you must make sure that you need to standardize the organism. Yeah, for example, what do you mean by standardize? You may want to standardize the same number of organisms if the sizes are almost the same, or you need to standardize their size. Okay, different size, different metabolic rate. Different size is not so good. The best way is different mass. If you can get different mass, it's even better. Always choose the better option if you can achieve it. But if you cannot achieve it, you choose the second best option with the apparatus that you have, okay? The type of organism. Type of organism means, for example, you got the same species of plant, but different variety. Okay, same species of plant, but different variety. So this is called the type of organism for the same species. You must choose the same type, okay? Choose the same age, choose the same species. So this is how you standardize the organism. Okay, up to here, do you all have any question? No, then we will move on, okay? Now we go to detailed procedure. This is where you need to pay attention to. When they ask you for detailed procedure for the experimental plan, you need to be familiar with whatever that you have learned in your AS and A2, whatever practical procedure that you have learned in your theory and practical. Whatever practical procedure that you learn in your theory and practical, you need to know so that you can write out the procedure, okay? For example, okay, how to calibrate the stage micrometer and eyepiece reticule, right? So you cannot, this is not about showing calculation. This is about telling a person how do you do it. So first, you must take a ruler scale, superimpose the scale of stage micrometer, right? Then after that, you need to find the 2 points of coincidence between the two scales, right? After that, you need to find out 1 division of your stage micrometer is equals to how many cm or how many mm of your ruler scale. So this is called the detailed steps. You need to actually spell it out very clearly. So therefore, you need to see whether you're able to write this very clearly, or else you need to go back and revise it until you can write it very clearly. Okay, so I already listed out most of them, most of those that you have learned in your theory, that the procedure you need to know for paper 5. How to prepare plant specimen to view mitosis and meiosis, right? So you have to take the root tip and you have to put into acid acetylocene. You have to heat it up at a hot plate for 7, 8 to 10 minutes. And then you have to dry it, and then you have to add, put it on a slide, add acetylocene stain, crushed it, okay? And then after that, view it, isn't it? So these are the steps involved, okay? Now there's one thing here I didn't mention is how to carry out biochemical tests like Benedict's test, DCBP test, okay, Burette test. So I didn't mention here. If you want, you can write it in your notes, okay, to make it complete. How to set up potometer, respirometer, and photometer, okay? How to get the leaf extract, and then how to set up and run chromatography. How to do electrophoresis. You will learn this in the next semester. How to immobilize enzyme into alginate beads. So this one you learn it in your AS, right? So if you've forgotten how to do it, please go back and revise. How to immobilize the enzyme, and how to identify. So this one, it is very interesting to see that in paper 5, they can actually ask slide question. It came out before, that's why I put it inside here. How to identify the structure of lungs, kidney, pancreas no need, flower need, ovary need, testis need, okay? Yeah. Maybe artery, vein also, so all the structures you've learned, you need to know, okay, how to, okay. Now these are the one in in your theory one. This one that you have learned in your theory. So all these are in your syllabus already already. Okay, so all these are in syllabus. So you need to know. Now the one at the bottom here, there are other usual procedures. Okay, other usual procedures means in experiment, what do you do in sequence? So step 1, I will do this, step 2, I'll do this, step 2, I'll do this. What do you do in sequence? Okay, for example, non-reducing sugar test. First, I must add 1 cm³ of my hydrochloric acid to the sucrose solution. Okay, then I'll heat it up for 1 minute, then I'll cool it down, and then I will neutralize it with sodium hydroxide. Okay, and then I will take 1 cm³ from here, and I'll add 1 cm³ benzoic solution, and I'll heat it up for 5 minutes, and then I'll check the color. So this is called the procedure, step by step. Step 1, what do you do? And then how much volume do you add? Okay, which solution to add in sequence, how much volume you need to add, how to set up the apparatus, how to set up your independent variable, how to set up your standardized variables, what are the precautions like, do you need to ensure that it's airtight? Do you know how to check that it's airtight? Okay, any wait time before the experimental start, okay? Time for temperature to equilibrate, time for pressure to equilibrate. Okay, these two are very, very important, or else you'll be doing at a different temperature, thinking that it's a temperature that you test. Okay, so all these are the important procedure. Okay, let me see. Why am my pen drawing things? Okay, clear. Okay, so accuracy and reliability is very important. Now, accuracy and reliability are two different things, so you need to know exactly what they are. Accuracy is not reliability. Okay, there are two different things, right? Remember, accuracy is how close is your result or data to the true value. Okay, how close is it to the true value? So this is called true value. True value means if you conduct the experiment exactly as what you have stated there, this is the volume exactly the temperature, concentration, the timing, everything exact, you will get this reading. So this is called the true value. But usually in experiment, we cannot get exactly to that temperature, exactly to the volume, because there is an error in all the apparatus that we use and also our own observation. So our results will be close to and rarely exactly on the dot, means true value. So accuracy is how close is your data to the true value. It's called accurate. So if you're asked, not if asked, how to conduct your experiment so that you can get accurate results. So these are the things you need to consider. Sometimes they ask you, so how are you going to improve the accuracy of your experiment? Okay, these are the methods here, all this one. Number 1, I'll skip first, I'll come back to this one. First thing to do is choose apparatus that give you better accuracy. Okay, for example, you want to measure volume. Okay, I've got so many apparatus I can use to measure volume. Which one is better? I've got graduated cylinder, I've got the beaker, okay, I've got a tip pipette, I've got a syringe, okay. So among these 4, which one is the best one I should use to measure the volume? Okay, I will use graduated cylinder. I'll use graduated cylinder, followed by a syringe, followed by beaker, followed by tip pipette. So you know that among the apparatus you have, do you have the best apparatus that give you the most accurate reading? Then you use that one if you have. Okay, if you don't have, what is the second best, third best? Okay, so, and if you want a constant temperature, it's best to use thermostatically controlled water bath. If your apparatus is too big, you cannot use water bath. You must use a room where the temperature is controlled, but you cannot say you use aircon to control. Okay, why? Because let's say you say, oh, I want a temperature at 40. You cannot use aircon to give you 40 degrees Celsius to control temperature. So examiner will not accept you say use aircon to control temperature. Just say control a room with controlled temperature. Okay. If you want to read the intensity or the darkness of a certain color, you need to use colorimeter. For example, orange color got different shades of orange, right? So if you use your eyes, it's all orange. The most you can say dark, darker, lighter, lightest. But how dark is dark, how light is light. So if you can give me the quantitative numbers, it's more accurate, correct? So therefore, we can use colorimeter better than our eyes to see and describe. Use pH meter versus using the pH paper. You know, pH paper cannot give you a better reading than the pH meter. So this is using instrument to give better accuracy. Weighing pen that give you 3 decimal place is more accurate than weighing pen that give you 1 decimal place. Okay, so apparatus. Then you can also do replicate and calculate mean. This can increase accuracy of your reading in what ways? I'll tell you. Let's say you got 3 readings, right? You got, I say 20, and the next one is 21, and suddenly the next one is 12. So when you got 3 readings like this, straight away you can see that 12 is an anomaly. All right. So because of that, when you calculate your mean, you will no longer take number 12 to calculate, because straight away between the 3, you already see that this one is not aligned with the reading of the rest of them. So you will actually remove it before you calculate the mean. So you only take these 2 to calculate mean. So when you take these 2 to calculate the mean, of course, your reading will be more accurate compared to if you also take number 12 to calculate the mean. So replicate and calculate the mean in this case will be able to improve the accuracy of your results. Okay, so that's why replicate is very important, and that's why replicate you must have got 3 readings, because you've got 2 readings, right? You wouldn't know which one is the correct one. Let's say you've got 2 readings, one is 20, one is 12. So hello, so which one should I use? I don't know. So I must have at least 3. That's why at least 3 for replicates. Okay, so you can remove anomalous before you calculate. Okay, then how to improve accuracy? Control all the control variables. Any other thing that can affect your result, you control them. Don't let them change. Okay, and then you can plot the graph of the best fit, also will increase accuracy of the reading. Because sometimes when you take a reading, your readings like this one, ding, 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 ding. Okay, 1 point here, 1 point here, then 1 point suddenly up here, then 1 point here. Okay, so maybe it is a smooth curve, so you draw the best fit curve. I wonder how to draw the best fit curve. Start from the first one, and then if it's a straight line, that means this is an error. But this cannot be error, right? Error must be like this, like this. Okay, so this one, chacat already. Okay, you want to draw the best fit curve. The distance between 2 points must be the same. The distance of these 2 points up above the line must be the same as the distance of this point below the line. It's called the best fit curve. Okay, plot the best fit curve will give you more accurate reading. So all the line along this best fit curve should be giving you a more accurate reading than your original data line. This is called the best fit curve. Okay, when you have the best fit curve, whatever line that falls on it will be more accurate. Whatever point that falls on it, let's say if this is the best fit, right, the blue color line. So whatever points that fall on it will be more accurate than your red color line. Any points on the red color line. So best fit curve. Okay, error bar. What is error bar? You learn it later. Okay, you can use the error bar to find out the accuracy of your reading. Okay, that one is statistic. Okay, so this is for accuracy, how to improve accuracy in your experiment. Now, how to improve, the next one is reliability. Reliability, I always memorize it as R, therefore it is R, repeatability. Repeatability means you measure 3 readings, 3 readings are very close to each other. You measure 5 readings, 5 readings are very close to each other. So this is called reliable or repeatable or valid or close to each other. Okay, so how to improve the reliability of your results? Here are the methods. Okay, First, of course, replicates. Replicates means you must repeat experiment, and at least you must have 3 readings, and only using the 3 readings, you calculate the mean, okay? And make sure you remove any anomalous before you calculate the mean, okay? So take 3 readings, remove the anomalous, and then calculate the mean, okay? 3 readings. Minimum 3 readings. Replicate, okay? 3 replicates. Next, you must allow time to equilibrate. Allow time to equilibrate can improve reliability. Let's say, for example, I want to conduct experiment 30 degrees Celsius, okay? So my enzyme must put into the water bath and for at least, let's say 5 minutes to ensure that the temperature of my enzyme is raised to the temperature of water bath, okay? So say this is my test tube and this is my water bath, right? Okay, and this is the water in it, right? So when I put my test tube into the water bath, I must allow enough time for the enzyme temperature to raise to the temperature of water bath, right? Then only when I do experiment, if it's 30 degrees, it's 30 degrees. So if you don't allow time to equilibrate, right, you just put inside and then straight away you use, right? This time I just put for a while, use. Next time it's 5 minutes I use. The next time it's 10 minutes I use, right? Okay, because you repeat experiment at different timing. The first one, 1 minute you use already. The second one, after 5 minutes you use. The third one, after 10 minutes you use. Then the first minute, maybe enzyme temperature is not 30 degrees yet. So therefore, when you take 3 readings, these 3 readings are not close to each other. So you must allow time to equilibrate to the actual temperature that you want. Okay, or the pressure is equilibrated for respirometer. All other factors must control. All control variables must be well controlled, okay? Plot a graph and draw the best fit curve and do standard deviation. Is a statistical analysis. Standard deviation, most of you have learned already, right? Standard deviation means, okay, the spread of the data around the mean, okay? Spread of the data around the mean. So let's say, for example, you got 5 readings, okay? You got 5 readings. Right, so you got 5 readings, you have 3, so you've got X1, no, your reading is Y, right? So Y1, Y2, Y3, Y4, and Y5. The one, let's say 3 enough, okay? So you got 3 readings, Y1, Y2, Y3, and then you calculate the mean, okay? So the mean is this value here. Mean, okay, can see or not? All right, so spread of the data around the mean since, say for example, if this is, let's say this is 20, your Y2 is 20, okay? Your Y1 is 18, and your Y3 is 21, 22, correct? Okay, so the mean of these 3 values is 20, right? So standard deviation shows the spread of your data around the mean. Means if you have got a graph, okay, and then your 20 is here, okay, so this is your mean value is 20 is here. Where are your data located? So you got 1 at 20, so you got 1 of the data is here, the second one is 18 is here, and the third one is 22 is here. Okay, what sound is that? Okay, so this is 18 and this is 22. So the spread of your data around the mean, the standard deviation is actually from here to here. So this is called the spread of your data around the mean. So this is called the standard deviation. Now let's say if this is your data, then you got another student that gets a spread. His data is, let's say, 16 and 24 lah, make it easy. So therefore, the standard deviation of the second student now will be actually further away. See not? right? Between 16 to 24 is actually further away from each other, right? Between this, okay, and this. So comparing student 1 and student 2, whose result will be more reliable? Then you have to look at the standard deviation. Of course, the first student result will be more reliable because they're more close to the mean. They're closer to the mean, so they are more reliable compared to the second student's result. It's further, the data are spread further away from the mean, so the repeatability is less because 16, 20, 24 is not repeatable. That means not very near to 20. So repeatability is less. That means reliability is lower. Okay, reliability is lower. Okay, so this is how you look at reliability of a result. Okay, and how to check reliability? You check standard deviation. Okay, now next thing is safety. For every experiment, you need to write at least one sentence in your experimental design or plan to talk about safety precaution. Okay, that means you need to identify what are the possible hazards for this particular experiment. And not only that, you must tell them, this one please add into your notes, you must tell them how to prevent from blah, 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 okay? That means you must tell the method of precaution. Okay, don't write how to prevent. Let's erase the whole thing away. Don't want to write how to prevent, right? Method of precaution. Okay, method of precaution. So what is the possible danger? What is the item that's dangerous? What is the action that's dangerous? And how to prevent it? Okay, you have to write like this, what and how. So the keyword here is what and how. What, how. Okay, what and how. So say for example, if you're using chemicals that are corrosive, like HCl, HCO2, H2O2, then you need to say that, so how are you going to protect yourself from this chemical? Lin Zhongwei. So what is the method that you will prescribe in your experimental plan for the safety precaution? Please do what? In order to prevent what? Lin Zhongwei.
We need to wear gloves, lab coat, and goggles.
Very good. Wear gloves, some more?
Lab coat and goggles.
Lab coat and goggles. Very good. Okay. So lab coat, we'll remove it because it is understood that must wear lab coat every time you do practical. So examiner will be looking for, okay, you need to wear gloves, and this is goggles or goggles. Goggles. Okay, wear gloves and to protect your hand and skin, and wear goggles to protect your eye just in case the chemical splash onto your eye or spill onto your skin. Okay, so very good. Thank you, Lin Zhongwei. I want to call Lin Zhongwei. Okay, thank you for your answer. Now, what about if you use any chemical that are very volatile, right? And then got very strong smell, then you want to conduct the experiment or handle the solution in the fume hood, okay, and switch on the exhaust fan. So this is something that you can write for that kind of chemical. Now this one is interesting. If let's say an experiment, you need to cut the potato or cut the root tip or cut the pond weeds. Why you want to cut the pond weeds? Make it shorter, okay? Now, how are you going to describe in your planning to somebody who's going to conduct experiment so that he will not cut himself? Okay, how to tell the person that to take the precaution so that he will not cut himself? Kaini, would you like to try this? How to prevent yourself from being cut? What do you do?
Put your hand further from the knife.
Approach your hand further from the knife. Okay, can you repeat again? What do you say?
Put your finger further from the knife.
Put your finger further from the knife. Okay. Okay. So you put your finger further from the knife when you hold the things and then you cut, right? Okay. So thank you for your answer. Yes. You can also say that you need, when you cut, this cut away in the direction away from your fingers. Okay. Cut the whatever things that you want to cut in the direction away from your fingers. That means if you, let's say you want to cut your potato here, okay, and your knife is up here. This is your knife, okay, and your knife, not like that one, okay, and your fingers are here, right? Ding, 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 ding. So you don't want your knife to be cutting this direction, right? You want your knife to be cutting to that direction, isn't it? Okay, so your finger will be holding something here, and this is something that you want to cut, and your knife, you actually slant it so that you cut it. How do I draw? You know the knife that you hold, right? Okay, you can slant it one, isn't it? Slant it to the left or slant it to the right. So when you slant it to the right side, this way, I see my hand, okay, your fingers, you're going to cut your hand, right? So you're going to slant that knife that way, right? So no matter how you cut, right, you cannot cut your finger one, okay? So you have to cut it this way, okay? Cut in the direction away from your fingers. Don't cut it this way, because you cut it this way, you might cut your finger. So this is slant the knife and cut it away from your finger, okay? So cut away from it. There are students who write, wear gloves. When you wear gloves, right, can you protect your fingers being cut by the knife or not? I think it's exam stress. I cannot think really. Okay. I think I've got students who write, wear gloves to prevent being cut. Okay, all right. Now, there's some apparatus that are connected to the electricity. So of course, you know that for this kind of apparatus, it cannot get wet. Okay, very important. And also you cannot handle it with wet hand. So how are you going to write for this kind of apparatus? Like, okay, you must make sure that your hands are dry when you handle it, especially when apparatus is put in the garden, right? You want to measure, you want to measure in the daytime, how many pollens, what are the pollen density in the air. You want to put this apparatus in the garden, open space, which is subjected to rain. And let's say you've got some pump to pump the air through it, so you're connected to electricity. So you must make sure that this apparatus must be fully covered and shielded from the rain. Okay, okay. So that is one precaution. Let's say if you're allergic, so you can write some more, right? You might be allergic to pollen. You may be allergic to pollen. So when you're conducting experiment that uses pollen, right? Allergy comes from you breathing in the pollen, right? So you want to wear a mask. You want to wear goggles to protect the pollen from coming into contact with your eyes, okay? And you want to wear gloves as well, that it will not come into contact with your skin that you might have allergy. So then you have to wear all this protective gear, right? Goggles, mask, and also gloves, especially biological specimen. Biological specimen means specimen from plant, specimen from animal, specimen from living organisms, okay? So you want to protect your skin from allergy, so you can do that. So you can write here, because this is just example, this is not complete, not the complete everything inside there, just example. Okay, so as you find more and more of them, you can add to your notes. This will be a very good notes for you for paper 5. Quite good one, huh? Okay, so after safety, the last one is control experiment. Okay, control experiment. Control experiment I've already mentioned to you in AS, so I will go through again one time as a revision. Okay. Now, control experiment is an experiment that you conduct in order to find out whether it is the independent variable that you have guessed in your mind that causes the result. that affects the result and nothing else. Okay, what do you mean by that? Now, let's say, for example, in your solution, you're testing whether the enzyme catalyzes the reaction or not. Okay, effect of enzyme concentration on the rate of reaction. So in this solution that you have, inside there, it's not only enzyme you have inside there, you also got water, because water is the solvent for enzyme. Okay, so when you do experiment and you get your result, so I know that we've got an enzyme and water, and I add a substrate into it. So I add substrate, okay, and then I will get product out from it. Okay, so that's what I expect in my experiment. So I put substrate into this, and then when I got enzyme and water, I will get the product. Now, but in this case here, there are two things here which may affect the result, because there are two variables in your beaker. There are enzymes and there are water molecules, right? So which is the one that actually causes the product to be formed? So therefore, when you have finished your experiment, you still want to do an extra experiment to verify that, okay, between these two factors that I have in my beaker, it is the active enzyme that result in the reaction, the product, not the water. So therefore, you do control experiment to show that it is the independent variable that you have identified as the one that actually affects your result, not the water. So therefore, there are two ways you can set up the control experiment here, correct? One is either you don't use the enzyme, you only use water, or you use the enzyme but you denature it, that means it's no longer active, it's the same, right? And then you see there are no products formed. So therefore, only the enzyme can result in the product, not the water. Okay, so control experiment. The second example here is a very good example to show that, okay, I'm going to do two tests to my orange juice. I'm going to do Benedict's test and DCPIP test. Benedict's test will tell me whether they are reducing sugar, and DCPIP will tell me whether they are vitamin C. So I want to find out. So are they reducing sugar? And then are there vitamin C, ascorbic acid? Here, vitamin C. Okay. So when I do the test, right, whenever I see it's positive, I say, oh, okay, this positive is due to reducing sugar. And then whenever I say it's positive for DCPIP, oh, okay, there are vitamin C because my DCPIP decolorized. Is it really true? We don't know, right? Because in this beaker, in my beaker here of orange juice, I have got two things inside there. I have got the, let's say, sucrose. I don't want sucrose. I have got glucose. And I also have got vitamin C, you know. I got two things in my solution, which is my orange juice. And then I do Benedict's test to this, it gives positive result. I do this very test, it gives me positive results. So are these positive results due to reducing sugar alone for Benedict's test and this very test vitamin C? I don't know. So I must do a control experiment to find out. So how do I do the control experiment? Okay, so in my control experiment, I must have got pure glucose solution and pure ascorbic acid solution. And then I will do the test to each of them separately. So I'll do Benedict's test, and then I also will do the DCPIP test. Okay, so after I do the test, I find, oh, Benedict's test, glucose gives positive. And then DCPIP, spelling wrong already, DCPIP, yeah, okay. This is, let's say, negative. Okay, very good. But ascorbic acid, Benedict's test give positive result. And then this one gave positive result. So now I found out that ascorbic acid also will give positive result for Benedict's test. So therefore, in my orange juice, when I have a 4+ in my result, plus, I know that it is all these plus are not only due to glucose, it can also be due to ascorbic acid that is present. So which means to say from my control experiment here to verify Benedict's test and DCRP, is it only due to glucose that give positive or both? Okay, then I will know that for this particular result is an overestimation of reducing sugar. So I know that based on the result for Benedict's test, the positive is not only due to glucose, but also ascorbic acid. So my estimation based on the color will be an overestimate because some of the color is not due to glucose. So that's the use of this control experiment. When you got two variables which may affect the result, you must test each variable separately. Then you know whether your result is overestimation or underestimation or correct. Okay. Now in this case, there's no underestimation. Okay. All right, so we take a break for 5 minutes. After that, we will come back and do the past year questions. So the past year question that we will discuss today is the one that I actually show you in your WhatsApp. This is also your homework to be submitted on Monday. So this is, let me see. This is Winter 11, paper 53, question 1. Okay, so if you have not got this paper, please go and print it out or retrieve it and put it into your iPad or your computer so that we can do this question together. Then you save you time to complete the rest and submit on Monday. Okay, so we'll take a break now for 5 minutes. So we will come back at 1:24. Okay, 1:24.